things to come. So the objective is your test is scheduled for a week from Thursday, correct?
Yes.
Okay, so your test is scheduled for a week from Thursday. It should cover synapses, muscles, and autonomic. Autonomic is a one-day lecture, so I'm not sure how much of it we're going to get finished on Tuesday. How far we get is where we stop the material, so I won't make you do it at home or anything, but it will get rolled over to the third test if we can't finish autonomic. So your test date is not going to change. It's still going to be Thursday of next week, which means you will have a short quiz on Tuesday because I want you to study muscle over fall break, right? Okay, so what we did last time was we got all the way through muscle contraction, and we briefly talked about muscle relaxation, right? Okay, so let's just I'm going to start with an animation and sort of review with you guys. Let me show you where these animations are. Okay, so if you go to, you guys have to stop talking. If you go to the bottom of the links page of melanieb.com, I have 4 animations in order that are all related to muscle contraction. If you watch them in order, it gives you a good understanding from the beginning to the end of what happens with muscle contraction. So let's just go here. Oh, no, not that one. Let's go to this one. And you guys are going to sort of just remind ourselves of what happens with Crossbridge Cycling and contraction, and then we'll go through relaxation briefly one more time. So as we are starting this, and we actually want to get this this sarcomere to contract, what has to be released in order to initiate this is what? Calcium. And calcium is, of course, going to bind to which protein in the sarcomere? Troponin. And when calcium binds to troponin, it will move the barricade protein called tropomyosin out of the way. Good. And then when that occurs, you can form a cross bridge, which is when the head on what protein? Binds to the active site on actin. Okay, here we go. And then I am going to start the power stroke. The power stroke is initiated when what falls off? Phosphate falls off. And then this one shows ADP falling off before the end, but it doesn't really matter. ADP is going to fall off. And now I'm stuck here until what happens? ATP binds, and that's going to release the cross bridge. And then hydrolyzing ATP will do what? Re-energize or reposition the cross bridge, right? So that's the whole Crossbridge cycling story. And you can do this over and over again. So now I want to talk about how you stop doing this. How do you make the muscle cell relax, right? So it's going to continue to do this until we start the relaxation process. And so we did this at the very end of last class, so let's do it briefly again. If I want to relax this thing, the first thing is you better stop doing action potentials on the axon, right? Okay, so you got to stop the action potentials on the axon. Or if you don't, you're going to continue to release what neurotransmitter? Acetylcholine is going to continue to be released. Okay, now for the acetylcholine that was already released, you need to get rid of it or it'll continue to stimulate the muscle cell, right? And the mechanism of getting rid of it is with an enzyme called acetylcholine esterase. So you've already seen the first part of this. This is the whole neuromuscular junction, voltage-gated calcium channel, acetylcholine release. And then what's going to happen is after it causes its postsynaptic cell response, then what's going to happen is there are enzymes that are present on the postsynaptic cell. And the enzymes are in the postsynaptic cell membrane, and it's called acetylcholine esterase. Okay, acetylcholine esterase will break acetylcholine down to acetic acid and choline, and then you reuptake the choline so that you can reuse it to make some more acetylcholine, right? So this is the mechanism of removing it from the synapse. Yeah. So that's the mechanism of removing it from the synapse. Acetylcholine esterase is easy to remember because if you look at the name, it's got the word acetylcholine in it and then the word erase in it, and the only thing between them is a st. So acetylcholine st erase. There you go. Acetylcholine esterase, so that the fiber will not continually be stimulated. Okay, so now I got rid of the acetylcholine, but what I'm going to have to do is, of course, that's going to stop the action potential, right? But what about the calcium that was released from the last action potential? What I'm going to have to do with the calcium that was released from the last action potential is put it back where I got it. Where did I get it? SR, but the SR is packed with calcium, so how do I get it to go back in there? I have to pump it, which requires ATP, exactly. So I'm going to pump it back in. And as you start pumping it back in, then of course the tropomyosin is going to fall back into position. And as the tropomyosin falls back into position, then you can no longer form cross bridges. Let go of enough cross bridges, and then what's going to happen to the actin? It's going to slide back into its long lengthened position, right? Does that make sense? So actin slips back into resting position, and the sarcomere is going to stay there until another action potential arrives. Is that good? Okay, so I'm going to go through a couple of clinical notes, and then we are going to go through that worksheet that I posted for you guys, and we're going to go through it as a group. Okay, so first clinical notes. What does rigor mortis have to do with this story? We briefly talked about it last time. The problem with being dead is that you are no longer making ATP, primarily because you are no longer taking in what? Oxygen, exactly. So no ATP is being made after death. So what is going to happen is I want you to think about how your sarcomeres will get stuck. So show me where your sarcomeres will get stuck with your hands. Yeah, they're not going to get stuck here. They're going to get stuck here, right? Because you actually need a new ATP molecule to release the cross bridge after a power stroke. So what's going to happen is if no ATP is present, you're going to be stuck in this position, right? In addition, you can't get rid of the calcium either, can you? Right? Because you can't pump it back into the SR. And then there's one other problem, although I don't generally talk about it. Calcium is in high concentration in the extracellular fluid, and it will start to slowly leak into the cell. So you'll not only be leaking it from the sarcoplasmic reticulum, you'll also be leaking it from the extracellular fluid. So you get this sort of slow and steady buildup of those initial power strokes. You're not going to get 20 power strokes, but you're going to get one over and over and over again in a bunch of different cross bridges. So what this is going to do is you are going to have sustained muscle contraction. You're not going to have so much that you generally wiggle and flop around, but you're going to have a sustained muscle contraction that builds up in the position that you died in. And so this guy right here. Okay, sustained muscular contraction. I want you to think about a living person trying to actually maintain that for any length of time, right? You would need so much ab and back and glute contractions in order to make that occur. If you could do it at all, you would be yelling, take the picture, take the picture. So this is rigor. This guy almost certainly, of course, died laying down, right? Obviously that picture wasn't supposed to be taken, but I figured you needed a picture of rigor mortis. So that's what rigor mortis looks like. And what happens is it will take a little while to build up, but it's temperature dependent because this is really all just diffusion, right? And diffusion occurs really quickly of calcium, right, at high temperatures and really slowly at low temperatures. So how do you predict how quickly you're going to go in and out of rigor? It's temperature dependent, but if it's at high temperatures, you may go into rigor in a few hours. You'll probably be out in about 10 hours. 12 to 15 hours is the average temperatures. You're going to be coming right back out of rigor at 12 to 15 hours. But if it's at really low temperatures, it sometimes takes 15 hours to get into it, right? And then you're going to be in it for a matter of maybe even a day or more, okay? So why would you ever start coming out of rigor? Because protein denaturation starts to happen, right? Your proteins fall apart, and as your proteins fall apart, then your cross bridges are going to let go because, of course, you're losing the structural stability of actin and myosin, right? So that's what happens. So with rigor mortis, sometimes, of course, people don't die in convenient positions like laying flat on their back. It's quite often they don't. Just so you know, one of the most common positions to die in is on the toilet, right? And that is in part, well, due to a lot of different things. One of them is the vasovagal response. We'll talk about this when we get to the cardiovascular system in more detail. But basically what happens is when you're straining to defecate, so you hadn't been eating enough fiber, right? And you already have heart problems, right? And you're straining to defecate and you actually put pressure on your vagus nerve, which slows down your heart. That won't kill most people, but if you already have cardiovascular problems, it can kill you. So a lot of people die on the toilet, which just So rude, really. When you die in the toilet, you die in this position or some variation of this position. And then it's kind of hard to get you out, especially if you have a teeny tiny bathroom. So this is like a series of bad events and bad decisions, right? I'm dying on the toilet. I'm probably fat, right? I got heart problems. I haven't been eating enough fiber, right? And maybe I'm in a little bitty trailer. And then add to that nobody is going to come find me for 24 hours, right? And then you have a situation in which they're going to have to get you out of the bathroom in rigor or partial rigor. Here's the deal with rigor. It doesn't let go. It is stronger than you are. It's very insistent, and generally speaking, they're going to have to break bones to get you out in that particular situation. It's not at all unusual for them to have to break bones. Your muscles won't let go. There's also other situations in which you can run into that, like in some religions and cultures, you're required to be buried within 24 hours. And no embalming. For instance, in Orthodox Judaism, you're supposed to be buried within 24 hours with no embalming. It probably makes sense to you that you're going to deal with a lot of people in rigor, right, in that situation. And a lot of times they'll have to break bones. If you didn't die laying down, they're going to have to break bones to get you into a casket. Go ahead.
How long does it take for that to set in? Sorry, you may have already said that.
It's temperature dependent. You're usually out of it in 12 to 15 hours. You're usually into it in about 5 or 6 hours. But it really depends on how hot it is outside, right? And you will go all the way through the stages of death really quickly if you are not in an air-conditioned situation and in the middle of summer. So you're going to go all the way to like putrefaction really, really quickly. These are, just in case you like wanted to know this, I just thought this was cool once I finally found it out. There are actually more stages than just rigor mortis. Rigor mortis is the third stage. There's pallor, algar, rigor mortis, liver, and then putrefaction, decomposition, and skeletonization. And that's months and months. But of course, temperature dependent, environmental factors dependent. But this is the one that we're talking about right here. So those are all those words, right? Don't you like use those in Trivia Pursuit? Okay, so that's rigor mortis. Let's talk about a couple of other things that are related to skeletal muscle contraction and skeletal muscle physiology. What I'm going to do is I'm going to tell you with each one of these what the problem is, and I'm going to ask you to try to predict the impact on the skeletal muscle fiber. You guys ready? Okay, poliomyelitis. Almost completely eradicated, but not quite. And what poliomyelitis does is the virus destroys the motor neuron. It kills it. And what do you know about your ability to regenerate neurons? Very little, slim to none, okay? So you destroy motor neurons. So what can you predict about the skeletal muscle fiber if you destroy the motor neuron? Paralysis. Temporary paralysis? No, permanent paralysis is what you have with poliomyelitis. So with polio, what happens is you have permanent paralysis, which of course is going to lead to the degeneration of muscle tissue, right? You're going to start losing your muscle tissue. And when you stop using your muscle tissue, you're going to start degenerating your bone tissue, right? So you're going to start losing both bone and muscle mass. Polio, here is what polio does. Polio causes paralysis and progressive degeneration of bone and muscle tissue. And the reason this one is worth talking about is because remember when you got polio? No, you don't, because there was a worldwide vaccination effort that almost completely eliminated polio. And it was effective, and it was vaccine-based, and this is a deadly and life-threatening disease. It was almost completely eliminated by a worldwide vaccination program. There's still little pockets of polio left in the world, and I'm afraid to know what's going to happen with this whole anti-vaccine movement. A lot of these communicable diseases that have almost been eradicated are going to have the opportunities that they didn't have before, which will matter to us because we're no longer vaccinated against polio. But I wouldn't doubt that at some point in the United States within the next 10 years there's going to be polio popping up because people are dumb. Go ahead.
So polio is a contagious disease?
Yes, it's communicable. Mm-hmm. Yeah, it's a virus. It's a communicable virus. But we don't get it any longer. Let me tell you a little success story. It's a little heartwarming success story about polio and polio vaccine. So Jonas Salk invented the polio vaccine it was the first vaccine, at a tremendous threat to his own health. He, for instance, vaccinated himself and his family to make sure that it worked. And then, unlike Martin Shkreli, the guy who decided to, my husband calls him Farmadouche, The Farmadouche, the guy who decided that he could take HIV medication and increase the price 720% for developing countries. Unlike that guy, what Jonas Salk did is he developed a polio vaccine and then gave it for free to the entire world to help eliminate polio. And that's why polio is really hardly been found in Western countries in forever. It's a couple of little pockets in Africa is what's left of polio right now. So anyway, it causes permanent paralysis because of damage to motor neurons. We had a president who had polio, right? Yeah, FDR had polio. He had partial paralysis. He was wheelchair bound at some points, but he had some strength. He could stand up. He was not stupid, however. He never debated or met with foreign leaders when he was in a wheelchair. He always did it standing. We tend to equate height and power, for better or for worse, and he was not unaware of that.
So were we all given the vaccine as a baby?
No, you weren't. Your prior generations were. You don't get the polio vaccine anymore. That's why it would be a big deal if it ever actually got into Western populations again.
My mom had it.
Yeah, my mom had it too, but I haven't had it. I haven't had the polio vaccine. It's been gone from Western civilization for quite a while. So we're no longer vaccinated against polio, and it still exists in the world. So that'll be an interesting one when they decide that they're not going to vaccinate their children and polio is in the United States. Don't even get me started on the anti-vaccine people. Oh, Orange County, I know. No kidding. There's a big, big fight in Orange County about vaccinating your children. I think it's totally fine to vaccinate your children as long as they don't hang out with my children at public school. That's totally fine. You have your own anti-vaccine school. You can have one over there. Okay, so that's rigor mortis and polio. A couple of other things. Cramps. What do cramps have to do with this story? What cramps do is usually because of an ion imbalance. What happens is you get progressive frequent stimulation of the neuron because of an ion imbalance. And of course, what's that going to do? Then the muscle is going to seize up. And when it seizes up to the point that it's really yanking on the connective tissue, it causes a cramp. That makes sense? Go ahead.
Are these the same as menstrual cramps, or is this different?
Menstrual cramps are hormonally mediated ion disturbances that will cause this. Yes. But menstrual cramps also, just the hormone itself will actually cause a frequent contraction. So the hormones cause ion disturbances, and the hormones also directly cause muscle contraction in the smooth muscle. Okay, so hypocalcemic tetany. Now we talked about hypocalcemia before, but now I want to just put it into the context of muscles. Remember when we talked about hypocalcemia, we said that calcium was the latch on your sodium channels, and if you lose the latch, then your sodium channels open inappropriately. So now I just want you to apply that to muscle. If that happens, you get action potentials when you didn't mean to get action potentials. If you have a very low concentration of calcium, that means that your muscles can get into tetanic contraction and you can't even relax them. That is usually due to a hormone imbalance and not reproductive hormones. So hypocalcemic tetany can cause just tetanic muscle contraction. Okay, so let's talk about muscular dystrophy. So muscular dystrophy is a genetic disease. It is X-linked, which means it is going to manifest more often in which gender? Males, exactly. Females have 2 X chromosomes. If I have a mutated single X chromosome, unless my parents were brother and sister, I probably don't have the same mutation on my other X chromosome. However, if I were a carrier for muscular dystrophy, all of my sons would manifest because I give them my X chromosome, right? That's where they get their X chromosome. So it's more common in males. There are females who have muscular dystrophy. Here's what it does. Muscular dystrophy causes a progressive degeneration of skeletal and cardiac muscle cells because, remember, your whole myofibrils attached to what part of the cell? The cytoskeleton. You have a weak cytoskeletal protein. So the structural stability of your cells is not strong. And every time you contract, of course, you're pulling on the cytoskeleton. It causes progressive degeneration of the cytoskeleton, of cardiac and skeletal muscle fibers. And exercise is not going to help the situation, right? And you can't really stop using your cardiac muscle, and you can't stop using your diaphragm. So what happens with these little boys is that they generally are going to, relatively young, die of respiratory or cardiac arrest. And there's some gene therapies for it. They're not commonly used. There's some things that they can do about it now. It's generally diagnosed in toddler years because there's a pull-up pattern that they have that's really different. So when babies start to try to walk, they have this pull-up pattern in which they start to pull themselves up and really use their legs to push their body up. Because they've got, decent leg strength like the rest of us. But with these little boys, what happens is they don't have very strong legs at all, so they tend to pull themselves up. So they tend to pull themselves up like this, like just directly, like holding their body straight. That's one of the diagnostics they use when they think a little boy might have muscular dystrophy. They don't use it as exclusively diagnostic because there is a genetic test that will confirm if muscular dystrophy exists. Their life expectancy is longer than it used to be, but they still don't usually live to adulthood, these little boys. And sometimes girls, just not very often. Okay, so myasthenia gravis. Myasthenia gravis is an autoimmune disease. So let me show you what this one does. Myasthenia gravis is an autoimmune disease. Your immune system makes a mistake and it starts to attack your own tissue. In this particular instance, what it does is it attacks the motor end plate. Destroys the motor end plate. So what's that going to do to the skeletal muscle fiber? It's going to paralyze it, temporary or permanent. It's not permanent because you're actually not destroying the cell. You just destroy the motor end plate. And as soon as your immune system gets distracted and goes to do something else, you will regenerate all of the receptors on the motor end plate. So what happens is this looks like sort of a roving muscle weakness. So it might manifest at first as like Bell's palsy, where one side of your face is weak. It might manifest as like a shortness of breath because your diaphragm is weak. You might limp for a little while if one of your legs is weak. You might actually lose motor strength in your arms. And it sort of roves around. Now, can it kill you? Yeah, because if it consistently attacks your diaphragm, it can kill you. So it can be life-threatening, but there's no predictable trajectory of it or any other autoimmune disease, really. You can't really tell how they're going to impact. And they actually sometimes do go in remission for long periods of time. But also, like all autoimmune diseases, there's no cure for it. So that's myasthenia gravis. And then the things that we talked about before, let's just put them into the context of this. Tetanus, what tetanus does is actually impacts the acetylcholine at the neuromuscular junction. And what it ends up doing is causing a continual stimulation of the muscle so that the muscle actually gets into tetanic contraction. So that's what tetanus does. And then botulism. Botulism kind of does the opposite of tetanus. What it does is it prevents the release of acetylcholine at the same location. And so what would botulism do to your diaphragm? Could you breathe in? No, your diaphragm contraction moves down when you breathe in. And so botulism can actually cause respiratory arrest because you can't contract your diaphragm. Does that make sense? So it causes problems with release there. So that's botulism. Botox Cosmetics, of course, uses that to a therapeutic or cosmetic advantage. So what Botox does is Botox, you don't actually use the living organism. You don't use Clostridium botulinum. But what you use is the toxin that you have generated in the lab. And then we take small quantities of the toxin and we inject it locally, not systemically, into an area. And what it will do is destroy your capacity to release acetylcholine at the muscle cell. And like I said, if you can't get this, if you can't do this, then you don't get those, right? So that's what Botox does. However, what it does is when you inject it directly in that area, it doesn't just destroy your ability to release acetylcholine for a short period of time. It actually degenerates your axon terminals, your cholinergic axon terminals, and it takes you 3-4 months to build them back up. So you'll have this temporary paralysis in the area, and that smooths out your wrinkles. You can use it in other ways too. Botox has actually been used to paralyze the muscles that tend to predispose people to migraines. Or they use it for hyperhidrosis, right? If people are sweating too much, you can actually use Botox here or hyperhidrosis on the hands. There's a bunch of different treatments that they've done using Botox. So anyway.
Are there long-term side effects of paralyzing?
You can permanently paralyze the muscle if you use Botox over and over again. Some people are trying to do that specifically so that they don't ever end up getting any wrinkles. If you do it repetitively, you will just end up sort of never moving those muscles enough to get any wrinkles. They're starting to do Botox on girls as young as 16 years old.
But I mean, like, in like a migraine aspect, like you don't want migraines anymore, so you paralyze.
They have not tried permanent paralysis with Botox, but it can do that. They're really careful when they actually use it therapeutically and how much and how frequently they use, because you don't want to damage something that's actually related to mobility in the process. But I mean, this is your frontalis muscle, and then these two right here. It's really not life-threatening if you paralyze those things, right? I mean, it makes you look weird, and everybody's like, hey, I'm so excited. So those are not life-threatening, of course. But my little plug is, if you're going to do Botox, please go to somebody who knows the anatomy of the face, right? You don't actually want to hit a nerve and end up with Bell's palsy, right? And you want to know where the muscles are that you're trying to paralyze. So I know that they offer, you can go for $300 and do it with wine and cheese. No, you need to go into an office with someone with degrees on the wall, preferably. Okay, so that's Botox. And then the last two are, we talked about Curare before, but what Curare does is it's an acetylcholine antagonist at skeletal muscle. So what's it going to do to the muscle fiber? Temporarily paralyze it, exactly. So the monkey will fall out of the tree so you can eat the monkey, or the wildebeest will fall down, or whatever the hell, or you can kill the person. And it's got a relatively short half-life, so it's going to be out of the tissue relatively quickly. Okay, and then the last one, nerve gases and pesticides. Some of the nerve gases that we developed, I know that as Americans we're in the habit of thinking that our human rights records are spotless, and that's comical. So we actually invented most of the nerve gases that are being used, or iterations of those nerve gases are being used. And the one that is used that has relevance to this story is that there's a nerve gas, I can't remember the name of it, I'll look it up before next time, that actually inhibits acetylcholine esterase. It's an acetylcholine esterase blocker. Not an acetylcholine blocker, but an acetylcholine esterase blocker. So what will that do if you block acetylcholine esterase? The muscle is going to continue to be stimulated. So here's how Curare would kill you. Curare would kill you because you couldn't actually inhale, you couldn't contract your diaphragm, right? But these nerve gases will kill you because you can inhale and you can't exhale, right? That's how those kill you. That make sense? Okay, some pesticides also do this. We didn't create the pesticides for that purpose, but that's part of the reason you want to keep them away from children, because they actually inhibit acetylcholine esterase activity. Okay, so nerve gases and pesticides. Okay, so now we are ready to move into skeletal muscle energy metabolism. Oh, wait, first we're going to do the worksheet. Forgot. Okay, so with the worksheet, what I would like for you guys to do is, if you want to do it on your own, you can, or get in groups of 2 or 3. And then here's what we're going to do. I'm not going to tell you guys the answer. You're going to tell each other the answer. And I'm going to start from the back of the room, and we're going to rove through. So take a minute, find someone who looks smart and friendly, sit next to them. You're going to have to separate. The whole wad of 5 of you is going to win the whole thing. She's a good person to have because she already did this once in the other class. No, this doesn't have anything to do with the anatomy story. It's only the Phys story. This is the Phys story that we told before. We're just doing it together as a workshop. Worksheet. Hey, Caroline, look at our shirts. You just ordered this shirt? I love the shirts. I bought my husband one, and I was like, oh, that's really cool. I'm going to go on to he's like, you just bought a matching shirt? Like, we're not going to wear them at the same time, don't worry. Okay. Who are you gonna get with? Over here? Okay. They're being shy. Okay, here's what's gonna happen. I'm gonna start back there with Caroline's group. And just so you can predict how this is going to go, I'm going to go this direction, that direction, work all the way down to the bottom, okay? Here's what I want from you. I want you to try. Because if you just sit there and don't try, then I am not going to have any capacity to give you a hint, to know what you're thinking, to reinforce you. So I want you to try. As long as you try, I'm going to be really nice to you. If you continue to not try, I will come back to you over and over again until you do try. Okay? Right? Okay. So if you will look at me and listen to me, I will give you hints. Okay? You ready? We're going to start at the top. Caroline's group. The functional unit of skeletal muscle fiber contraction, the thing that contracts, look at me, is called a what? No, not you guys. Caroline's group. Sarcomere, exactly. Okay, now over here, you guys, Jill and Wad that's with Jill. Okay, the theory that explains the skeletal muscle contraction is called the blank blank theory, and it is the only theory we have talked about the entire semester. What is it? Sliding filament theory. Good. Okay, so now we're going to go to the next one, and what we're going to do is we're going to start the way that we started the story. We have to start at the nervous system, right? Then we go to the muscle cell. After we get it to contract, we'll get it to relax. So the first part of the story is trying to get the muscle cell excited, right? Okay, starting with the nervous system. So you guys that are next to Jill, you two right there, yes? So here's what we're going to talk about. The action potential is going to arrive at the end of the axon on the somatic motor neuron. And what's going to happen on the somatic motor neuron is the blank, blank, blank channels will open on the synaptic terminals of the presynaptic cell. So you guys. Voltage-gated calcium channels will open on the synaptic terminals of the presynaptic cell. Okay, C. I can't see who's in the middle. Somebody with a beanie on. Yeah? Hi. Okay, so we are at C. Blank is released from the synaptic terminals. What do you release from the synaptic terminals to stimulate skeletal muscle? You guys? Are you guys together?
They are now.
What's released from the synaptic terminals? Strange? Acetylcholine is released from the synaptic terminals into the junction. Okay, so I don't know if you guys are together, I can't tell. Yeah? What is the junction called? It's what a synapse is between a neuron and a muscle. It's a blank junction, neuromuscular junction, exactly. Okay, it's released into the neuromuscular junction. Okay, we are at D now. We just released it, so that shouldn't be hard. What did we just release? You guys right there? Acetylcholine, okay, binds to, what does acetylcholine always bind to? Just generally, what does acetylcholine bind to?
Receptors.
Yeah, acetylcholine binds to receptors, yes. On the blank, blank, blank portion of the muscle cell membrane. So look at me. So you already did it, and you're next. So what's that curvy part, right? On the muscle cell membrane that has all the receptors on it. You ready? Uh-huh. The curvy portion that has all the receptors on it.
The
Sarcoplasm.
Sarcoplasm is the cytoplasm. Doesn't start with sarco. How about you guys? Do you know it? Yeah, motor end plate. Good. Motor end plate of the muscle cell membrane. That's the motor end plate. You good?
Why wouldn't, like, I was originally thinking, like, okay, 3 blanks is going to be postsynaptic neuron. Yes.
No, because it is actually a part of the muscle cell membrane. Motor end plate portion of the muscle cell membrane. Okay, good? Okay, E. That was you guys. So E is you guys in the middle, right there. Right there. I'm looking at you. So E says these protein receptors are also acetylcholine-gated what kind of ion channels? So right behind you. Acetylcholine-gated what kind of ion channels? This one, E. Sodium. These protein receptors are also acetylcholine-gated sodium channels. Okay, you guys are next. in the middle. Opening these channels causes a blank polarizing graded potential. Yeah, they got an easy one. A depolarizing graded potential and excitation, and it's even easy to spell, excitation of the sarcolemma. So G, do you want me to skip you?
I already did it.
Okay, so G, over to you guys. If threshold is reached, the action potential begins on the sarcolemma and travels deep into the muscle fiber via little blank, big blank. Say it. T-tubules are good. Okay. H says, now the action potential arrives. Are you guys ready? Over here. The action potential arrives at what? You go from the T-tubules to stimulate the what? Sarcoplasmic reticulum. Good. Action potential arrives at the sarcoplasmic reticulum. Okay, you guys are next. Action potential causes what to be released from the SR? Calcium to be released from the SR. Whew, that was way less painful than the 8:30 class. Okay, back to the back. Caroline, it's you again. Yeah. I know. Okay, action potential causes blank to be released from the SR. Calcium, and now we're at B. Calcium binds to the sarcomere at a small protein fiber called what? Troponin. Good, with confidence, yes. C. Jill, back to your group. With calcium bound to troponin, the barricade protein blank. Tropomyosin, which prevents crossbridge formation, is lowered. Okay, now at D. Right after Jill, blonde in stripes. So the blank heads can now contact the blank active sites. Which protein has the heads? Not you guys. Myosin. Which protein has the active sites? Actin has the active sites. When the heads and the active sites are bound to one another, it is called a, move over to you guys in the middle. Crossbridge. Good. Okay, so now we are at a crossbridge. We're at E. To initiate the power stroke, right after you guys, blank must fall off. Phosphate must fall off. You got it. To initiate the power stroke, the phosphate must fall off the what head? Which protein has the heads again? Everybody? Myosin heads. Myosin heads now pivot for the first power stroke. At the end of the power stroke, let's do you guys right there. ADP. At the end of the power stroke, ADP falls off the myosin head to prepare for the next cycle. Crossbridge detachment. You were hopeful, huh? Crossbridge detachment. Over to you guys again. Which protein has the heads again? Myosin. The myosin heads must release the, which protein has the active sites again? Actin, active sites, for another power stroke. And this requires, don't yell it out, this requires you guys in the middle. ATP, good. This requires ATP. For a sarcomere to shorten, this attached power stroke detach cycle must happen many times. This repetitive process is called, whichever group didn't answer the last time. What's it called? Crossbridge cycling, exactly. It's called crossbridge cycling. Okay, now we're going to get into the relaxation phase. Okay, it's very relaxing doing this, isn't it? In order for the muscle fiber to relax, so listen to what we're talking about. In order for the muscle fiber to relax, the action potential on the blank must end, or acetylcholine will continue to be released. Let's do you guys. In order for the muscle fiber to relax, the action potential on the blank must end, or you will continue to release acetylcholine. Presynaptic cell or nerve cell or axon, right? Anything that implies neuron instead of muscle cell.
A little space. What answer?
I don't care about memorization. I care about learning.
She needs to have everything.
So I will never tell you the answers to those questions. Okay, so the action potential on the axon must end, or acetylcholine will continue to be released. Acetylcholine that was already, this is you guys, over here. You guys. Acetylcholine that was already in the neuromuscular junction must be destroyed, or the muscle receptors would continue to detect it. The enzyme acetylcholine esterase destroys acetylcholine at the neuromuscular junction. How do you spell it? Acetylcholine esterase. That's how you spell it. Okay? No acetylcholine means no depolarizing graded potential on the muscle cell, and therefore no, this is you guys. If you don't get a depolarizing graded potential, you don't get a what? That's fine. I'll read it again. C. No acetylcholine means no depolarizing graded potential on the muscle cell, and therefore no blank blank follows on the sarcolemma. You don't have a depolarizing graded potential, you don't have a action potential. You got it. Action potential follows on the sarcolemma. Okay, D. No AP on the sarcolemma means that blank, say it together, will not be released from the sarcoplasmic reticulum. Calcium will not be released from the sarcoplasmic reticulum. Good. Calcium present in the sarcomere must be removed for, raise your hand if you know this one. Calcium present in the sarcomere must be removed for the blank barricade proteins to go back up. Back up. Raise your hand if you know it. Go ahead. Tropomyosin proteins to go back up. This is accomplished by pumping calcium back into the SR and requires ATP. Yes, good. Now, the blank thin myofilaments. Raise your hand if you know which ones are the thin ones. Actin. Thin myofilaments slip back into position, and the sarcomere lengthens. Woo! You did it. But I know you hate me, but let me ask you one more time. Which protein has the head? Which protein has the actin side? Actin. Okay.
Oh, man.
You have no idea how much better you did at that than the 8:30 class, honestly. Well, it's really hard for them. They're like hardly conscious. It's really like, it's kind of hard. It makes sense to him. He did take anatomy from me, but I didn't do any of this in anatomy. No. Hyperhidrosis?
Wait, no, I actually did Botox stuff in your armpits for that.
You did?
It's the max dosage 6 times and it didn't do anything.
Are you kidding?
But I'm gonna like keep going and then I'm just gonna end up like it's so nerve-wracking when your doctor is just like, oh yeah, I've never had this happen. Let's just increase it. Like, okay, wait.
That sounds like a good idea. Okay, so we're changing gears. We are done with muscle contraction at that level. We are going to now back up a little bit and talk about where you get all that ATP. Okay, so before we get into how the ATP, how we get it, let's talk about the places in this whole story in which ATP is necessary so that we can get an appreciation for how energetically expensive that story is that we told. Okay, first off, when the neuron is having the action potential, does that require any ATP? Yeah, an action potential actually requires a boatload of ATP, especially for a repolarization process. How about exocytosis of acetylcholine? Does that require ATP? Yeah, exocytosis always requires ATP. And now I'm going to have an action potential along the sarcolemma, and that's going to require ATP during the fixing of it. Okay, then the release of calcium doesn't require any ATP. How about crossbridge cycling? Boatloads of ATP. And then if I ever want to stop crossbridge cycling, I'm going to need to get rid of the calcium, right? And that requires ATP as well. So this is an incredibly energetically expensive process. And so what we're going to go through is how the skeletal muscle gets its energy. But in addition to needing a lot of energy, it also needs to be very responsive very quickly, right? Because your skeletal muscle is what keeps you from getting eaten by a tiger or hit by a bus, right? It's the quick responsive skeletal muscle. So what we need is we need to make sure that it can always respond very, very quickly, whether I've eaten anything recently or not. So there are lots of like plan A, plan B, little check mechanisms, little savings accounts in skeletal muscle. So let's start a skeletal muscle. And let's say, okay, let's pretend for a second that if I wanted a skeletal muscle to contract, I would have to, for instance, eat a Snickers bar, wait for it to break down, get absorbed, get all the way to the cell. You know you would die, right? You would get hit by the damn bus if you had to wait for that to occur. There's all kinds of little savings mechanisms. So let's talk about what they are in order. The first one is just how you left your myosin heads from the last Crossbridge Cycling. Don't you always leave them in the high energy position so that you will get this crossbridge formation and one power stroke for free the next time? I mean, it's not free, but you paid for it a while ago, right? And you know what happens with something you paid for a while ago. It feels like it's free, right? Okay. So you get this and you get that, right? That's what my husband, I was like, just put the boots in the closet. And he's like, where did you get those? I was like, oh, ages ago. That was like a $300 that I spent a long time ago. I don't even know why I care. We have separate bank accounts, but I still like fib about it. Oh no, I've had these forever. I have no idea. He thinks it's hilarious too. He's like, I don't care. I know, but I feel the need to lie. Don't know. Okay, so creatine phosphate is the next thing. So that's only going to get you one power stroke, right? Because if you don't have ATP, you can't do another one, right? Because you can't release, right? So you get that first power stroke for free. But then what is your next quickest access to ATP? Your next quickest access to ATP, let's look at this figure. Now, we could do all of this. That's not very quick, right? All of those processes to get ATP. You guys with me? Your next quickest access to ATP is to use your used ATP, which is ADP, in the cell, okay, and quickly rephosphorylate it by a one chemical reaction. So what happens is inside a skeletal muscle fiber, there is creatine phosphate, okay? And creatine phosphate holds onto a phosphate. And here's what it does. Bless you. Here's what it does. It can quickly rephosphorylate ADP so that now you have ATP, exactly. Does that sound like this would be faster than waiting for this to happen? Yeah. So this is quick access to ATP. It is present in the cell membrane. You have enough creatine phosphate to get you about a minute of skeletal muscle fiber activity. Okay? So you got enough for about a minute, and then you're just out of creatine phosphate. Okay? You're going to reform it later after you get finished with skeletal muscle contraction. So hopefully that got me out of the way of a bus. But what if it's a really insistent bus, right? I need more than a minute of activity. So what's next? What's next is, fascinatingly, when you start using your skeletal muscles, do you give your cardiovascular and respiratory system a heads up before you start doing it, or you just start doing it, right? You just start doing it, right? And then your cardiovascular and respiratory system are like, hey, wait a minute, let me catch up. So your respiration rate has to increase and your cardiovascular blood flow has to increase, but your skeletal muscle has to act before they do. So here's the deal. The skeletal muscle has some little piggy banks stored so that it can always act really quickly. And here's what it does. So the next thing is that even before the cardiovascular and respiratory system can keep up with the oxygen requirements, remember that I had small quantities of oxygen stored inside the cell on myoglobin, right? And that means that even before I'm supplying enough oxygen, I can do a little bit of aerobic cellular respiration, right? That makes sense? So I've got a little bit of oxygen stored inside the cell, so I've got a little bit of aerobic cellular respiration before the oxygen catches up. And then in addition, remember that your skeletal muscle fiber also stores glycogen, right? So now I've got a little bit of glycogen, a little bit of oxygen. That means I can start doing aerobic cellular respiration before my cardiovascular and respiratory system catch up. Does that make sense? So this glycogen that's stored inside a skeletal muscle fiber, this is its piggy bank, right? It is going to pull on that piggy bank. It doesn't share with any other cells, and it doesn't have to wait for blood flow to get to it. So it has instant access to it. So you can start the cellular respiration process. As you're running out of your creatine phosphate, you're starting this process, right? And then by the time this gets going, I am going to have access to, for this breakdown of glycogen that occurs inside skeletal muscles. Depending on your skeletal muscles, depending on what you've eaten and what you've stored, that can last for about 5 to 10 minutes of skeletal muscle activity. So now we're about 11 minutes in, max, right? So we had 1 minute with creatine phosphate, right? and we've got now about 10 minutes with stored glycogen. Hopefully that's an adequate time for your cardiovascular and respiratory system to catch up, right? And so now let's say my cardiovascular and respiratory system catch up, and then what I start burning is, instead of stored glycogen, I start burning glucose. And hopefully I've also started delivering adequate quantities of oxygen to do so, right? So now this part, when I'm burning blood glucose, depending on what you've eaten, right? Depending on how much glucose you had stored, this can last to about 30 minutes. So now I'm about 42, 43 minutes in. Good? Okay. So blood glucose breakdown by glycolysis and oxphos, that's about the next 30 minutes. And then after you have done this, most of us are going to be out of blood glucose by that point, okay? Now, if you've been carbo loading and that kind of stuff, you can make it last a little longer if you've been paying attention to that kind of stuff. But most of us, after about 45 minutes, are going to switch over to fatty acids. We're going to burn fatty acids in our bloodstream. We save those for after I've run out of glucose. So you're going to start burning fatty acids. Now, does that mean that you have to work out for 45 minutes to lose any fat? No, because this is happening at the cellular level, okay? What I'm saying is this cell has 45 minutes before it starts burning fatty acids. But over the course of any day, if you wiggle more energy out than you take in, you are going to burn fat to support your body-wide metabolism, right? But to force a skeletal muscle fiber over to burning fatty acids, it usually takes about 40-45 minutes, depending on. Does that make sense to you guys? Okay. So after that, the energy is primarily from fatty acid breakdown. Now, go ahead.
That's happening at the cellular level, and but then if you keep exercising after that, are there other ones that are starting up later? So then those are.
Other tissues have entirely different predispositions. Other tissues will start burning fatty acids much more quickly than skeletal muscle will.
Okay.
But this is just for a skeletal muscle story.
Like 3 hours.
Oh, your whole body's going to be burning fatty acids if you're running for 3 hours, right? But your skeletal muscles have about 45 minutes of their activity, right?
So after 45 minutes, it's not like they each start at a different sequence.
Well, they do kind of. You're going to see a little bit later in this set of notes that there's different metabolic characteristics to different kinds of skeletal muscle fibers. So kind of hold on to that question and maybe we'll readdress it. Okay, so, but there's one more part to the story, and that is that skeletal muscle will still try to get you out of the way of a bus even if you don't have any oxygen, because skeletal muscle has the capacity for anaerobic cellular respiration, and anaerobic cellular respiration will kick you back through glycolysis to make a little bit more ATP. It's not going to get you a lot of ATP, but if there's a bus there, it's pretty damn useful. But it generates lactic acid or lactate as a byproduct. So let's look at that for just a second. Okay, so remind yourself of the things that you know. This is just adding a little bit of a tweak to the story that you already know. If you take glycogen and break it down to glucose, it's called glycogenolysis, right? Take glucose and break it down to pyruvate and ATP, it's called glycolysis, right? And then the rest of the story usually goes this way. Take that and continue through Krebs and oxphos, and you get boatloads of ATP and then also carbon dioxide and water. So we know that story. So what is new about this story? The new part about this story is that skeletal muscle, not all tissues can do this, but some other tissues can as well, can take that pyruvate plus a little bit of ATP and kick it back through glycolysis via this process that we call anaerobic cellular respiration. And it generates slightly more ATP than it uses, and it also generates lactic acid as a byproduct. Okay, so this is your like plan Z, right? I ran out of everything, but I still need to get out of the way of a bus, and your skeletal muscles can do this, okay? So they are going to generate lactic acid as a byproduct. And we'll talk about what people think about lactic acid and what's true and not true in just a second. Okay, so let's just acknowledge which of these processes require oxygen and which don't. So this one doesn't require any oxygen, right? And this one doesn't require any oxygen. It produces small quantities of ATP, but nothing impressive. This one doesn't require any oxygen, but still produces small quantities of ATP. And it produces lactic acid as a byproduct. It was thought for decades that lactic acid was responsible for muscle soreness, and then they figured out that it's actually not responsible for muscle soreness. It is actually responsible for muscle fatigue, in addition to the fact that you don't have very much ATP, which of course is going to make your muscles fatigue relatively quickly. Apparently the lactic acid, the current thinking is the lactic acid actually impedes sarcomere contraction, like physically impedes sarcomere contraction as it builds up. but it's not responsible for muscle soreness. And they had to come up with some decent experimentation before they could figure that out. So what if, at the same time that I do an activity which is generally going to cause muscle soreness, I infuse the muscle with massive quantities of oxygen, right? Then it is not going to go into anaerobic cellular respiration and therefore not generate lactic acid. And then I assess muscle soreness afterward to see if muscle soreness is still there. And lo and behold, muscle soreness was still there. If you suffuse it with oxygen and there is no lactic acid buildup, you're still going to get muscle soreness. The muscle soreness seems to be because of connective tissue strain. But why did they assume that it was lactic acid? Because it's correlated with the high levels of lactic acid. But correlation does not equal causation, right? So they assumed forever that the lactic acid buildup was related to the soreness, but it's not. It's maybe related to muscle fatigue. And they did some separate experimentation to show that. So your trainer is still going to tell you that your lactic acid buildup is related to muscle soreness, but it's not. It's generally there at the same time. Now, what tends to help with both muscle fatigue, lactic acid buildup, and muscle soreness is to increase blood flow to the area to help flush out the lactic acid, which reduces the muscle fatigue, and to help increase blood flow to the area to actually repair after connective tissue strain. So you can see how you got that sort of miscommunication, in which like those two things seem to always travel together, right? So, but they aren't actually directly related to one another. So that's the relationship of lactic acid and muscle. Okay, but of course, plan A would be that you didn't do that, but you did this, right? You actually did, you had plenty of oxygen. Go ahead.
Why do people supplement with creatine?
Oh, that's a good question. So let's address that. So you can buy supplements in the health food store or wherever that are creatine or creatine phosphate supplements. And so what do those do and are those useful? Okay, so creatine and creatine phosphate supplements. If you are doing an activity that one more rep would cause demonstrable gain, then creatine or creatine phosphate might get that for you. So for instance, if you were a power lifter, and you could get a couple more seconds of activity, and that one more rep might actually end up building more strength, right? Because you've got a little bit more ATP. Now, if you're a cyclist, is creatine phosphate going to do anything for you? No, it's really not going to do anything for you. So here's the deal. Creatine phosphate does seem, and remember this is a nutritional supplement, so it hasn't been evaluated by the FDA, right? That's not how nutritional supplements are evaluated. It does seem to increase muscle mass. But the other thing is it's also sort of giving you a false positive because it's also super, super water soluble, and it makes you retain water as well. So you gain weight, but some of it is probably water weight gain. It's also kind of hard on your kidneys to supplement creatine phosphate, so you have to really increase the amount of water you're taking in to do that. And the reason that I would be cautious if I were a person who was trying to use creatine phosphate is that skeletal muscle is not the only place that creatine phosphate is used. And we don't know what high levels do to other tissues, other precious tissues like cardiac muscle, like the retina of your eye, and gentlemen, like your testes. Creatine phosphate goes to the testes as well. I don't know. I wonder like 25 years in the future if we're going to see like testicular cancer, testicular whatever, or blindness associated with all of this creatine supplementation. So I always say if you're a grown person, you can do whatever the hell you want. But if we get into situations in which like high school football coaches are saying you need to like take creatine supplements, I would be the worst parent to have as a football mom. First of all, my kid is never going to play football ever, right? Because like this is like arguably the most important part of you, right? But second, you've met those people. That is not the sharpest knife in the drawer, generally speaking, right? I mean, every once in a while you get a like brilliant one, but it's just not the thing, okay? And they're like telling your kids. I mean, how many coaches do you think have actually taken college level physiology? No, they haven't. Some of them take exercise phys. Go ahead.
So in high school, my friend was talking about the argument that it may or may not be a steroid.
No, it's not a steroid.
So why would that be a conversation?
Because people don't know words. That wasn't very sensitive, was it? No, there are smart football players. There are. I mean, but just let's look at statistical anomalies. There's statistical anomalies. Okay, so I'll teach everybody what a steroid is. Oh my goodness. I'm just like, you know, nastier than usual after the presidential debate. And I was like, that word does not mean that. That doesn't mean that either. Okay. My husband is like, you really shouldn't do that and drink at the same time because your children are not going to like you. Okay, so, so enough. All right, so now let's talk about what happens after you stop working out. So let's say you were using your skeletal muscle fiber. Here's one of the things you might have heard. Have you heard that your metabolism stays high for a while after you stop exercising? And of course, you have also heard or experienced the fact that immediately after you stop exercising, your cardiovascular rate does not go down instantaneously, neither does your respiratory rate. Is it because you can't slow them down that quickly? Yeah, you can, but you don't want to because you still have a lot of work to do. So let's see what kind of work you have to do. First off, the oxygen that was bound to the myoglobin inside the cell and the oxygen that is in the hemoglobin in your blood vessels all needs to be replaced. So that requires high levels of oxygen for a decent time period afterward. Does that make sense to you? So you have to put it back, which means you need to continue to take in large quantities of oxygen. The creatine phosphate has to be remade. So we have to do this process. We have to take creatine plus ATP and make it back into creatine phosphate so that I can have quick access to those phosphates as soon as I use my ATPs, right? So that means I need a bunch of this, even after skeletal muscle contraction, right? Now, I don't go this way during muscle contraction. I go that way after muscle contraction. I go this way during muscle contraction. So do you see that we're going to need high quantities of ATP even after we stop working out? Yeah? Okay, and then what else? The lactic acid that I generated, you can make it back into pyruvate and ultimately go back this direction, but that is an aerobic that is an oxidizing reaction, so it needs oxygen as well, okay? So that requires oxygen. And so replacing all of these things that you used are either oxygen requiring or energy or anabolic reactions. And so your metabolism needs to stay up for a while. So you continue to breathe deeply and rapidly even after you stop exercising. Your cardiovascular rate stays up for a while after you stop exercising. They say that the purpose is to repay the oxygen debt. They call it the oxygen debt, but it's not just oxygen. It's also nutrients and, you know, energy molecules as well. And also remember, you probably heated up your body, right? And you have to cool down your body as well. So your metabolism stays up for minutes to hours, depending on how much you worked and which muscle groups you worked after you work out. So that has good and bad things associated with it. Have you ever tried to work out and then go to sleep? It doesn't work for me, right? I'm like all wound up and have to think about everything for like 2 hours. But if you work out in the morning, what tends to happen is you tend to have a more active day because you actually get your metabolism up and you tend to just accomplish more things. So people who work out in the morning, not because they burn more calories, there's nothing special about morning, but they tend to, over the course of a day, end up using more energy because you actually got started and you got going and your metabolism was high and you just get a bunch of crap done. You know, if you get stuff done in the morning, you tend to have like an effective day the entire day. What was your question?
Were you talking earlier when we were talking about circadian rhythms, were you talking about time to exercise afternoon time?
Yeah.
So why is that the case and compared to what you're saying now?
I don't actually know. There must be, I wonder if it's nutrient level availability or oxygen availability. I don't think it has directly to do. Yeah, I don't know that answer. That's a good question. I'll think about that one. Okay. One last little tidbit about this is that this whole energy conversion from ATP to movement is inefficient, like all energy conversions are. And you only actually end up sort of capturing about 40% of the energy to use for movement. The 60% is lost as heat. So muscle contraction tends to heat up the body. And that's either a byproduct of something that you were doing, or you can use it as shivering to heat up the body on purpose. You're using this sort of inefficient muscle contraction. Go ahead.
Why does your body feel more energized after you worked out if it's doing all that?
Your metabolism is higher for hours after. So you tend to feel energized when your metabolism is higher. Like all of your cellular processes are working slightly more quickly. Go ahead.
Well, I think everything would be more oxidized, right?
More oxidized, more blood flow to everywhere, including your brain, right? Pretty much all of those things are going on after you work out.
Are there other chemicals that are released that make you feel really
Oh yeah, You release, that's a CNS thing, but you tend to release endorphins. I think some people release dopamine when they work out. Mine doesn't. Mine's like whatever the opposite of dopamine that's like, oh God, is this really necessary? Is this what I'm doing with my life? Right? You're like walking over and over again and not going anywhere. I'm like, oh my God, this is giving me existential angst. It is. I'm sure. Yeah, you guys go do those studies. Oh, yeah, from doing outside. If I can do anything that doesn't actually feel like I'm working out and working out simultaneously, riding my bike, hiking, any of that stuff, I'm sure that all kinds of happy chemicals released. But if like Fox News is on at the gym, no, I'm not releasing any dopamine. That's not what's happening. It's not happening. Okay, so now let's talk about single fiber muscle contraction. You did this stuff in lab. You are having a test next week?
Yeah. Same Tuesday alone, Same week.
Oh. What? I'm not moving. Slab schmab. 80%, 20%. I win. They can move it. Okay, so let's talk about the mechanics of muscle contraction because you guys did this in lab. So here you go. Okay, so remember looking at this thing in lab, right? This is the response of a single muscle fiber to a single stimulation, right? And so what you're going to do is you're going to stimulate the muscle. If it were inside me, I would stimulate it by releasing acetylcholine. If I'm in the lab, of course, I'm going to zap it with like electrode to get it going. But what's going to happen is you end up with 3 phases. You've got the latent period, you've got the contraction phase, and you've got the relaxation phase, right? Okay, so this latent period, this contraction phase, and this relaxation phase. Before we go any further, what I want to do is put together the story that we told about what's happening inside a muscle and between the neuron and the muscle together with this diagram, okay, to make it relevant to the stuff that we do in lecture. Ideally, lecture and lab end up going together. In Phys, there's a lot of problems with that, but let's make it work for us at least this once, okay? So, first off, between the stimulus and any measurable tension generation in the skeletal muscle fiber, there's some crap that needs to happen, okay? So I want you guys to tell me what's happening. So I'm going to tell you, I'm going to get you started. Right here is where I actually generated a depolarizing graded potential on the muscle cell, okay? And here is where I'm actually generating tension. So help me figure out what has to happen in between there. Okay, start.
Okay.
You can see why there's a latent period. That's why I'm having you guys do this. So what has to happen? I got a depolarizing graded potential on the muscle cell. And then what? Sodium channels. And then what?
And then the voltage gated.
Voltage gated channels. And then that's an action potential. And then the action potential has to do what?
And then the potassium.
T-tubules, right? And then what? You were right. Huh? Then release of calcium. And then what does calcium do? Binds to troponin. Tropomyosin moves out of the way. All of that before I've even formed a crossbridge, right? And now I form my first crossbridge and start a power stroke, right? And so as I form my first crossbridge and start a power stroke, I'm about right here. What is happening during the contraction phase? Yeah, crossbridge cycling, right? Crossbridge cycling, crossbridge cycling, crossbridge cycling. As long as you have ATP, it's going to be happening, okay? So now let's put it together with the relaxation phase. Now I want to relax. In order to do that, what do I have to do? You have to stop, but you can't just stop. You have to do what? You have to clear out the calcium. You have to pump it back into the SR. And as soon as it releases, your tropomyosin goes back into position. As soon as you get enough of that happening, then actin slides back, right? So this story that you told in lab is directly related to the story that we've been telling in lecture. It's just that in lecture you go into a hell of a lot more detail, right? That makes sense? Okay, so with that in mind, this, by the way, just to reiterate, this is not what happens in a whole muscle. This is in a single cell. Okay, we're going to try to get to whole muscle contraction in just a second. So now let's talk about a couple of different things. Okay, so before we get to how whole muscles contract, I need to define tension and load and talk about how they're related to one another. So tension is the force that is exerted on any object by a contracting muscle. So if you contract your abs with me right here, just contract them, okay? You're generating tension. So microscopically, you are doing sarcomere contraction, are you not? But you're not doing enough sarcomere contraction to overcome the load, right? So if I want to overcome the load with this muscle, the rectus abdominis, this is what I would have to do. I would have to generate enough sarcomere contraction that I overcame the load. Or if you look at it like this, right? Here's the biceps brachii. Generate enough sarcomere contraction so I can tense my biceps brachii without actually wiggling. So microscopically, of course I'm wiggling. But if you don't generate enough tension to overcome the load, then you won't actually have any measurable contraction, right? And you can get to the point where your tension is just not adequate to overcome the load. So if I come over here and I try to use my biceps brachii, I'm generating tension, but I can't overcome the load, right? Okay? So the relationship between tension and load is this. If the tension can overcome the load, then you're going to wiggle, right? If the tension can't or doesn't overcome the load, then even though you are using sarcomeres, you are not going to be moving. Okay, does that make sense? Okay, so our capacity for tension overcoming the load is different from person to person, right? So if I compared me to him, he would probably have more capacity for his tension to overcome the load because testosterone increases actin and myosin. And so you're going to actually have more sarcomeres with testosterone, right?
I would put your money.
So, well, I'm 5'2", okay? And I haven't been to Pilates in at least 3 weeks. So you're probably going to take me. So, but that can change over time, right? Your muscles can generate more tension, less tension, those kinds of things. We'll talk about how you increase your tensile strength in your muscles as we go through. So that's the relationship between tension and the load, and they oppose one another. So now let's start talking about whole muscle contraction, and you talked about these types of contractions when you were in lab. Okay, so again, when I did Phys, we actually took the muscle out of a critter and clipped it to something and did this, and you guys just get to simulate it. Okay, so here is what we're going to simulate here. We are going to have a muscle which we are going to attach to a rigid support. And by rigid, I mean that the muscle could not possibly overwhelm that support, right? You're going to make it out of solid metal, and you're going to use a muscle that doesn't have the capacity to generate that tensile strength. And then what I'm going to do is I am going to stimulate that thing. Internally, of course, the stimulation would happen with acetylcholine, but you can open voltage-gated channels without it. So you can stimulate it externally. And what you're going to do is you're going to record the tension generated in the muscle, right? So this is a whole muscle. Even though this looks like a twitch, it's not a twitch because this is a whole muscle. So notice what happens is you generate tension, but you do not change the length of the muscle, right? Okay. So this type of contraction is called an isometric contraction. What does isometric imply to you? Same length, exactly. So what you are doing with isometric contractions is, although at a cellular level you are generating a contraction, what happens between the tension and the load? Yeah, the tension is not or cannot overcome the load. So when are you doing isometric contractions? You're doing them right now with your neck, right? To hold your neck still, right? That's an isometric contraction. If you contract your abs but don't try to wiggle your body, you're doing an isometric contraction. That makes sense? Writing?
Yeah.
Writing is isotonic contractions. Yeah, isotonic contractions. If you do yoga, you do a hell of a lot of isometric contractions during yoga. You get yourself into a position and then you hold that position. It's an isometric contraction. Might have been isotonic before, and then it's isometric when you're holding it. So isometric contractions are really common. They're more about postural stability than they are about wiggling you around. Do they still use ATP? Yeah, they still use ATP, right? You don't need to generate as much force, as much tension, because you're not trying to overcome the load, but they still generate ATP. So that's an isometric contraction. And now let's do a different type of contraction. This one is called an isotonic contraction. And with an isotonic contraction, you have to set it up a little bit differently. Here's your relaxed muscle. Here's a load, but it's on a movable lever. Okay? Now, how much of the load? Well, you're going to have to experiment with that to figure out how much tension the muscle can generate to overcome the load. With this one, if we experiment around with a little bit, and we've got a movable lever, we can stimulate this muscle, and the tension can overcome the load. Do you see what's happening there? Okay, so now I want to look at two different measurements. The last time all I did was measure tension because the length didn't change, right? This time I am going to measure tension, and I'm also going to measure the degree of shortening. Okay, so what happens here, and you've experienced this, it just probably won't sound very relevant until I describe it to you. What happens here is I am generating tension, and then once I get up to the tension is adequate to overcome the load, instead of continuing to generate tension, I'm going to use it for movement, right? So if I try to do this, right, my biceps brachii is generating tension. When it gets adequate, it actually overcomes the load. Does that make sense to you guys? So what's going to happen here is if you measure tension, it goes up and then it plateaus because instead of generating more tension, I'm using that for movement, right? So now let's talk about the name of this one, isotonic. Same tone, same tension, because do you see once you get up to the adequate tension, it plateaus? So that's why this one's called an isotonic contraction. But isotonic contractions are wiggling, right? You're generating enough tension to overcome the load, so you are actually going to wiggle. Is that good? Okay. So examples of isotonic contractions. Yeah, any kind of movement, walking, running, using your pen, any of those kinds of things are isotonic movements. You're generating enough tension to overcome the load. Good? Okay, so one last type of contraction that you didn't cover in lab, but it's relevant to this and it's interesting to think about. So they call it a lengthening contraction, which totally sounds like an oxymoron, but I can make it make sense to you. So if you've ever done a back bend, like since you were 10, you know that you don't just flip over because you are definitely going to break something. You just flip over. So when you do a back bend in yoga, what you do is you like engage your abs, right? And then what you do is you pull the origin and the insertion away from one another. And so you are contracting the muscle, but you are lengthening it simultaneously. Okay? Primarily, you're doing that by using the antagonistic muscle pair, right, to move against it. And you're trying to generate more tension with that one than you are with this one. So that's called a lengthening contraction, but it's really just a subtype of one of those two. Which one?
Isometric.
Yeah, it's a subtype of isometric contraction. It's an isometric contraction here because I'm engaging it and I'm not trying to overcome the load. But on this one, it's a what kind of contraction? Isotonic, exactly, right? So you're opposing those two, right? And it wouldn't be, just so you know, if you just like flip backwards, it doesn't mean you're doing an isometric contraction here, right? Only if you actually engage the muscle and start crossbridge cycling and then stretch it, then it's actually a lengthening contraction, which is an isometric contraction. Does that make sense? Right? Why the hell would you want to do that? Because it stabilizes your joints. That's why you would want to do it. If you are doing an activity that is really hard on a joint, you want to engage the muscles around the joint to protect the joint, right? And that's why you just don't just flip over and do a back bend in yoga. Okay? All right. Okay. So, you guys are all right with those? Okay, so we will stop there for today.